subject of this presentation is the nervous tissue. It is one of the four basic tissues that makes the human body. It has special features, and we will discuss these features in this presentation. What is the theme of this PowerPoint presentation? The theme is critical thinking. What is critical thinking? That means I look at an image, get facts out of this image, put it in my mind, think of it, and then criticize it. What is the process of criticism? It is the procedure of questions and answers. I have facts on the image. I ask myself and answer these questions. What are the parts of the nervous system? We have the central nervous system and the peripheral nervous system. Now, what is the central nervous system? It is the part of the nervous system that is present inside the skull, which is the brain, and the part present in the vertebral column, which is called the spinal cord. Now, these two structures are the main center of activity and integration. Now, the peripheral nervous system is made of peripheral nerves, and we will have a look at the structure of peripheral nerve. And the peripheral nerves come out either from the brain, and then they are called cranial nerves, or come out of the spinal cord, and they are called spinal nerves. Looking at this situation, I can see that there are very large cells. These are the building blocks of the nervous system, and they are called the neurons. And there are few of them in the field of this slide. Now, beside these neurons, there are very large number of supporting cells for the neurons, and they are called neuroglia. And inside this yellow circle, for example, you can see there are so many dark spots. These are nuclei of the supporting cells, the neuroglia. And if you compare the size of the neuron to the size of the neuroglia, you can see that the neurons are so many times larger than the connective tissue of the nervous system, which is called the neuroglia cells. This is a section of the neuron, the basic unit of the nervous system. And is this neuron the same as other cells? The answer is, well, let's look at its components and then we will answer this question. First of all, we can see that this cell has a cytoplasm. cytoplasm. This cytoplasm, when it is in a neuron, it has a special name called perikaryon. Now, why is it so blue? Because it contains so many of the organelles of any cell. It contains mitochondria, it contains Golgi apparatus, it contains an endoplasmic reticulum, and let us stop at the endoplasmic reticulum. The endoplasmic reticulum in a neuron is rough endoplasmic reticulum, and it produces lots of RNA molecules. These are called Nissl granules, because neurons, they are supposed to develop a chemical substance that will deliver the impulses to other cells. These substances are called neurotransmitters. So Nissl granules are very good identifying feature of a neuron. So the cytoplasm is perikaryon. Then we have the nucleus. Now, before putting the circle, you can see the shadow of the nuclear membrane. The nucleus is large. Because the cell is large, the nucleus is large and very easily identifiable. Now, inside the nucleus, we can see the compact circular dark structure, which is the nucleolus. Now, do we see the nucleolus in a neuron quite easily? The answer is yes, we can. Can we see the nucleolus in other cells very clearly? The answer is no. Then we can see that coming out of the body of the neuron. Is there another name for the body of the neuron? The answer is yes. It is soma, S-O-M-A. And coming out of the body of the neuron, we can see that there are processes that come out of the cell body of the neuron. And there are several types of these processes. There are the dendrites, as we can see here. There are several dendrites, and in addition to the dendrites, we have the axon. The axon is the process of the neuron which is going to send information out of the neuron to other cells, while the dendrites, they collect information and bring this information into the neuron. Now, how do we know that this is an axon and not the others? We look at the very beginning, the cone-shaped beginning of the axon, and we can see here that it is not containing the blue material, the Nissl granule. This sign is telling us that this is the axon, the outgoing process, and there are no Nissl granules in this structure, the beginning of the axon, and this area devoid of Nissl granules is called the axon hillock. Now, just like in the previous slide, the neuron is very large cell, and the neuroglia, or shortly the glial cells, the connective tissue cells of the nervous system, are small cells compared to the neuron. another details of the different parts of the neuron, the basic building block of the nervous system. Why another example? Just so that our eyes and brain becomes used to this structure. This is the cell body, or the soma, and the cytoplasm of this neuron is the perikaryon. Then we can see the shape and the outline of the nucleus, although it is faint, and inside the nucleus we can see a very evident nucleolus, and inside the perikaryon we can see these dark spots. They are the missile bodies. They are RNA particles. Then the neuron also contains microfilaments to keep its shape. We have dendrites, several of these structures. Now, why are these two dendrites? Because their beginning contain the missile granules, the blue color. Now, this is an axon. Why this is an axon? Because it does not contain the blue color, the missile bodies. And this is the beginning of the axon, and you can see microtubules, and if we look for the missile granules, they are not there. And the small nuclei around this neuron are nuclei of neuroglial cells. This is a third example of a neuron, the classical building block of the nervous system, and we can see that this is the soma, the body of the neuron, cell body, and we can see that the nucleus has we can outline the periphery of the nucleus, and we can see the very evident nucleolus, and this red arrow is pointing at an dendrite, and this is another dendrite, two processes coming out of the body of the neurons, and this is what? Guess what? No missile granules, therefore this is an axon. This is an outgoing pathway for the nervous stimulus. What can I learn from this image? First of all, now I am used to identify this cell as a neuron because of the body contains the dark spots, the missile granules. I can see the nucleus very well. I can see the nucleolus very well, and this cell is very much larger than surrounding cells, and this is an axon, the outgate of the nervous signals. And why this is an axon? Because it does not contain the missile granules, and if we follow this axon, the question arises, are axons going to divide? The answer is yes, they do. How many branches do they usually have? The answer is they usually have very few branches. So this axon is going to continue, and it gives a collateral branch of the axon. This is another example of the nervous tissue, but it is part of the peripheral nervous system. It is in the what is called dorsal root ganglia, something we will learn later on. And of course, what are these very large cells? The answer is they are neurons. Can you see the nucleus very well? Yes, I can. Can you see in this neuron the cell, the nuclear membrane? The answer is yes, I can. Can you see the nucleolus? Yes, I do. And are there granules? The answer is yes. There are many granules inside the pericardium. So this is another example of a neuron present in the peripheral nervous system. Very short conclusion is that nervous tissue is made of what? It's made of neurons and glial cells for support. Now, regarding the structure of a neuron, usually the starting point is studying the cell body or the soma, and we need to learn more about the neuron. How do we learn more? By asking questions. You ask questions, you learn more. You don't ask, you are not learning very well. Are all neurons of the same type, same structure, the same pattern? The answer is no. So this slide is going to tell us what are the basic types of neurons. Now, in the figure to your left, the rounded structure in the middle is the soma, and the dark circle inside the soma is the nucleus. Now, in this neuron, we can see that on one side of the body there are several processes. They are short, they are highly branching, and they join the soma. These are called dendrites, and they collect information from different neurons and feed the information into the cell body. Now, this is the axon. The axon is the outlet for the information from the neuron. You can see it is long. It has a cover made of several cells, and at the end of this axon you can see it has few branches. So this neuron has several points of receiving dendrites and one axon coming out of it, and this is going to describe as multipolar neurons. Then we go to the next. We can see the soma, we can see the nucleus, but we are going to look at the processes. The processes that are attached to this body of the neuron. There's a process going up, okay, and at the end of this process it is going to branch extensively, and these branches are short. Now, when we look to the process going down, we can see that it is the axon, the process that comes out of the neuron. The conclusion is this: the cell body is giving two branches, each one at a pole. So this neuron is called bipolar neuron. And we go to the next. Identify the body of the neuron, the cell body. Identify the nucleus, and then we look at what is attached to this body. What is attached to this body? It is one structure pointed by this arrow, and from this short structure a process goes up and at its end branches extensively. Therefore, these are the features of dendrites. At the other end, we can see the long process, which is the axon, ending by dividing into few branches. And in fact, this neuron has two processes, but they have united together to form a single process coming out of the neuron, and therefore this is called pseudo-unipolar neuron. Is it really unipolar? The answer is no, but it looks like. Therefore the word pseudo means it looks like. Then we go to the last neuron to your right, and we can see that this cell body is going to give one process coming out, and then this process, as it comes out, it is called unipolar neuron. Now we need to learn more about the neuron, and we learn by asking questions and trying to answer these questions. Now here is a question for you. On what basis are neurons classified? What is this image telling me? It is telling me that the orange cells have a body and a nucleus, and it is multipolar because these two neurons, they have multiple dendrites and then one axon coming down. So now we know what are these two neurons. Now we go and see the glial cells, the connective tissue cells, which are of different types. Here is the blue cell, and it's called oligodendrocyte. Oligo means few, dendro means processes, and cyte means cell. We can see that this cell is going to send arms, and these arms, they are going to catch an axon and puts an envelope on it. There are four processes, and each process is enveloping small part of an axon of a cell, and it is giving a cover to another axon of another cell. So this is an oligodendrocyte. Then we have this green cell, which is having processes all around the cell body, and when you look at it and see what it does, it holds things together and in a fixed fashion. So it sends processes to the covering of the axon, and it sends processes to the dendrites to keep them in place and support them. Then we have this cell. It's a small cell. It is the smallest, and it has multiple processes, and these processes are dividing. Therefore, this cell is going to be called microglia. Micro means small, microscopic, and glia means the connective tissue cell of the nervous system. This is a phagocytic cell, the cell that goes around in the nervous system and clear things. Here is a 3D image of a cell, the yellow one, and we can see that there's a cell body and there are many processes coming out, and we can see that one process is coming to the axon and giving it a cover for a short distance, and therefore this is what? This is oligodendrocyte, the cell that covers axons in the central nervous system. This is a good exercise for you, and I will leave the process of asking questions, answering them, and deciding what type of this cell is. Now, axons must be insulated from the surrounding area. What was the cell in the central nervous system that insulated or covered part of the axon? The answer is oligodendrocyte. Now we move to the peripheral nervous system, and it has lots of axons. Are these axons insulated? The answer is yes. How are they insulated? Is this insulation done by oligodendrocytes? The answer is no. Then what makes this cover? The answer is Schwann cell. Now, this Schwann cell is going to produce what is called the myelin sheath, and the myelin sheath is made of several small segments with a small area between these segments. We will come to it very shortly. So the starting point is that a Schwann cell comes to a small part of the axon, surrounds the axon, and then starts turning around and around and around till it finishes by producing a sheath to the axon. The axon is in the middle, and the sheath is made of several layers of the plasma membrane of the Schwann cells. So this is what is called the myelin sheath in the peripheral nervous system. This is the real image, the real world image. It is a transmission electron microscope that magnifies things thousands of times, and we can see here an axon which is covered by a very thick dark black area. This is an axon with a myelin sheath, and there are several examples of this myelinated axons. If we look into the this structure inside the red circle, we can see that the cell producing the sheath has covered the axon, but it has not turned round and is not producing several layers. So this is not myelinated axon. This situation is present in the central nervous system, and the myelin is produced by oligodendrocytes. This is an electron microscope image of a cross section in an axon, where you can see that the light area in the middle is the cytoplasm of the axon, and you can see that the outer cover, the myelin sheath, is made of several turns of a Schwann cell. This is a 3D drawing of the myelin sheath of an axon, where you can see that there are 3 different small segments of the myelin sheath. Why 3? This is only a small area, and if the axon is long, how many of these segments make the myelin sheath? The answer is very large number of these structures, and you can see that this is, for example, in the peripheral nervous system, this is a Schwann cell. And you know why I know this is in the peripheral nervous system? Because I can see that there is a nucleus of this cell surrounding the axon. In case of the central nervous system, I don't see a nucleus, because what makes the myelin sheath is a process of oligodendrocyte. And this is another Schwann cell, and look at the area between the 2 Schwann cells. It has no myelin sheath, and this is called node of Ranvier. In the high magnification of a light microscope, we can see this is a cross section of a nerve, and we can see the series of Schwann cells, and an area between a Schwann cell and the next is an area which has no cover by the myelin sheath. This is called node of Ranvier. Here is another example. Now, this is a 3D image of a cell which is called oligodendrocyte. Where do I find oligodendrocyte? The answer is in the central nervous system. Can I find it in the peripheral? The answer is no. And this oligodendrocyte is concerned about what? It's concerned about axons. Now, what does it do? It produces part of the myelin sheath of an axon in the central nervous system. And here is an axon running inside a part of the myelin sheath. Here is the oligodendrocyte, the cell body, and this is part of myelin sheath, and this is the other part of myelin sheath given by this cell. Now, between these 2 parts of the myelin sheath, pointed at by the black arrow, these 2 segments of the myelin sheath, they are not coming from this oligodendrocyte. They are coming from another oligodendrocyte. So here is the question: does an oligodendrocyte produce myelin sheath of a single axon? The answer is no. Oligodendrocyte sends processes to axon number 1, 2, to axon number 2. Now, number 1 is going to have segments of its myelin sheath from oligodendrocyte number 1 plus oligodendrocyte number 2 plus number 3, and so on until the whole axon is covered. Now, as we have went through several lectures, and the theme of all the lectures was asking questions, and as the time of the exam approaches, the more you ask questions, the more you learn. Now, you ask the questions and you look for answers. Now, the issue of questions and answers is the basic principle of what? It's basic principles of the exam. You get a question and choose the answer. How nice it is if you get a question that you already asked yourself and you looked for an answer. So the more questions you ask yourself, the better you do in the exam. What is this image telling me? I can see that there is a neuron on the top right part of the image, and there's a capillary, a blood vessel running across the image, and here is one type of microglia called astrocyte. Astro means star-like structure. It has a body and has multiple processes. Now, we had the idea that astrocytes is a connective tissue cell special for the nervous system, and it has processes attaches to different structures to keep them in place and give them support. If we look at this image again of astrocyte, we can see that this is the body, and these processes like this one is coming out of the astrocyte and then approaching part of the myelin sheath and holding it in its place. Does it form any part of the myelin sheath? The answer is no. It only supports it. Now, here is a blood vessel, a capillary, and we can see that this capillary is also covered by a part or one of the processes of astrocytes. So it supports the blood vessels and shares in the formation of what goes out of the blood vessel, a part of its control, and this controlling system is called blood-brain barrier. So astrocytes forms part of the blood-brain barrier. Now, the story of what goes on in the nervous system is this. Dendrites get messages from different neurons, collect them, and send them to the cell body, the soma. The cell body is going to process this information, and it decides what to do and sends this decision through the axon to the next neuron. So this process is going to take place in a structure called the synapse. It's a point of communication between an axon of a neuron and a dendrite or a cell body of the next neuron. The story starts like this. The nerve impulse comes through the axon, reaches the structure that is called the presynaptic axon, last part of the axon, and then it reaches a dilated structure at the most end of the axon. This is called the synaptic knob. Now, this synaptic knob contains vesicles. That these vesicles contain neurotransmitter, usually acetylcholine. This vesicle is approaching the end of the synaptic knob, holding the neurotransmitter. Then the wall of the vesicle is going to fuse with the plasma membrane of the last part of the axon, the synaptic knob, and opens to the interstitial space, and the neurotransmitter is released from the synaptic knob. This space is called synaptic cleft, and these neurotransmitters, they are going to be picked up by receptors of which cell? Of the next cell. And this is the way the neuron transmits its messages to the next through the structure of synapse, by the way of releasing neurotransmitter. The question is, does the synaptic knob, the last ending part of the axon, does the synapse produce the vesicles and the neurotransmitter? The answer is no. It is produced higher up in the soma, and they are carried down to the synaptic knob. Now, the synaptic knob, which is going to go through the process of neurotransmission, it needs several things. They are provided by microtubules, and there is lots of mitochondria, because this process needs energy. This is an image of a peripheral nerve. Now, the peripheral nerve is covered as overall by connective tissue, which is called epineurium. Now, in what way are the axons arranged in a peripheral nerve? The answer is in the form of fascicles. What is a fascicle? A fascicle is a bunch of axons. So this bunch of axons is called a fascicle, and this fascicle is covered by its own connective tissue called the perineurium. Connective tissue that covers a group of axons is the perineurium. Then each single axon is covered by myelin sheath, of course, plus a very thin layer of connective tissue called endoneurium. So what is this image telling me? This image is telling me that peripheral nerve has connective tissue elements. The one covering the whole nerve is the epineurium, and the axons are not arranged in a one-way fashion filling the whole peripheral nerve, but they're arranged in bundles. These bundles are called fascicles, and each fascicle is having its own connective tissue cover. That is the perineurium, and a single axon has two covers. One is an insulator, the myelin sheath, and on its outside a thin layer of connective tissue called endoneurium. Now we have collected so many data from many images. Now it's about time to write down our notes. Everyone, when he or she sees images, study them, and at the end must hold a pen and writing the notes on the paper. If you do that, you discover what you know and what you don't know. Neurons. There are 4 regions in a neuron. Cell body, the soma of the neuron. It is going to integrate information. It receives information from dendrites, and the nucleus is surrounded by cytoplasm, and this cytoplasm is called the perikaryon, and this cell body is going to contain most of the organelles: mitochondria, Golgi apparatus, endoplasmic reticulum, and so on. On one pole of the cell body we have dendrites. They are the receptive portion. They collect information and send it to the cell body, and it is the only way the body is going to get information. On the other pole we have axons. The axon leaves the body and goes to other neuron or other target. It is the conductive portion of the neuron, the messenger that sends the information from the body to other structures. The efferent signals. The word efferent means the sending part, the sending signals flow down the axon, goes through the axon, and in the axon signals travel in one direction. There is no coming back, so there is no back and forth. It is only forth. When they flow signals in one direction, they go and end up in the terminal branches of the axon, and this axon can be very long, can be up to a meter long, while the dendrites are very short. The axon hillock, the cone shape very beginning of the axon, uh, which contains no nestle granules, and that is how we identify a process as an axon. And then the initial segment, the part of the axon very near the cell body, is called the initial segment of an axon. It is a structure of an axon which attaches to the axon hillock. Then at the end of the axon we have synaptic terminal, the synapse. It is the end of the axon and it carries information from the neuron to the next neuron, and this synaptic knob or synapse has high concentration of vesicles which contain the neurotransmitters. Now, we have seen all these information. This is part of the mind map of the basic tissues. We have discussed the other arms of this mind map, and we are looking now at the nervous tissue. One of the very important features of the nervous tissue is that it is excitable. You stimulate it, produces an action. The other excitable tissue was the muscular tissue. This nervous tissue is made of nerve cells. They are the neurons, and they are also supporting cells, and in the central nervous system these supporting cells are called glial cells, and we have seen the different parts of glial cells. Now, this nerve cell, the neuron, has a cell body, one axon, and several dendrites. The cell body has a large nucleus and nestle granules. Now, the axon is usually long, dendrites are short, and there are types of neurons: the unipolar, the bipolar, the multipolar, the pseudo-unipolar. Then this neuron has to send its information to the next neuron and through the synaptic communication. And this synaptic communication is a process of conducting signals from one neuron to other neuron or other neurons, and it is made of synaptic knob, the last part of the axon, synaptic gap, and then the postsynaptic blade belonging to the next neuron or neurons. We have three functional classifications of neurons. There are sensory neurons, neurons that connect the periphery of the body into the central nervous system, like feeling the temperature, feeling the pain. These are sensory neurons. And there are motor neurons, neurons that coming out of the brain or spinal cord, their axons, they go to effector organs like muscle or a gland. And there are interneurons. Interneurons, they connect, they are short, and they connect one neuron to the other. That's why they are called interneuron. This slide is another summary of what goes on in the parts of the neuron, and I'm going to leave you to view it at your rest. This is a comparison between axons and dendrites, and I will leave it to you to study it. Now, the nervous system usually has two types of looking. Part of the central nervous system looks gray, and it's called gray matter, and it is made of nerve cell bodies, dendrites, axons, and glial cells. The collection of these structures gives a gray color called gray matter. As in this image, you can see that the outer part of the brain is gray and the inner is white, while in the spinal cord, the section to your far right, the arrangement is opposite. The gray matter is in the center and the white matter is on the outside. Now, what is the white matter? The white matter is made only of axons and glial cells. Okay, so there are no cell bodies of neurons in the white matter. Again, neurons are of different types. There are motor neurons, carry impulses from brain to skeletal muscles. Sensory neurons carry sensations from skin to the brain. Interneurons, they communicate between sensory and motor neurons. We have another kind of arrangements. If there's a cluster of neuron cell bodies in the CNS, the central nervous system, then this is equal to nuclei. We find some nuclei in the white matter of the brain. Now, if there's a collection of neural cell bodies outside the central nervous system, in the peripheral nervous system, then this arrangement is called ganglia. The singular is ganglion. Now, it's about time you test your information and your understanding. So this is a slide that is going to be left to you to give your conclusion. understanding.